You felt a craving 30 minutes ago. You don't know why. You assume it's your brain, your hunger, your choice. It may not be. Inside your gut right now, over 100 trillion bacterial organisms are alive, feeding, competing, and signaling. They outnumber your own cells. They manufacture over 90% of your body's serotonin. They produce the chemical precursors to dopamine. They synthesize GABA, the brain's primary calming neurotransmitter. And they communicate with your brain through the longest nerve in your body, sending signals upward around the clock. The craving you felt may not have originated in your mind. The anxiety you carry may be the chemical signature of a microbial community you never chose. You are not simply driving yourself, you are an ecosystem, and your passengers have significant influence over the wheel. What follows is one of the most unsettling discoveries in modern neuroscience. Your gut contains a nervous system of its own. The enteric nervous system spans the entire length of your gastrointestinal tract, from esophagus to rectum. It contains approximately 500 million neurons, more than the spinal cord. This network is so complex and autonomous that neuroscientists call it the second brain. It can operate entirely independently of your central nervous system. Cut the nerve connecting your gut to your brain, and your gut keeps digesting, contracting, and signaling without any input from above. The enteric nervous system was described in 1921 by John Newport Langley at Cambridge University. But the idea that this second brain might be shaped by microbial inhabitants was not seriously investigated until the 1990s. Your microbiome is the collective term for the trillions of microorganisms living in and on your body. The gut microbiome alone contains at least 38 trillion bacterial cells by recent estimates, exceeding the number of human cells in your entire body. These bacteria carry a combined genetic library containing roughly 150 times more genes than the human genome. They belong to thousands of species, dominated by two major phyla, Firmicutes and Bacteroidetes. Their composition is shaped by how you were born, what you have eaten, what medications you have taken, and the people you have lived with. No two people have the same microbiome. It is as individual as a fingerprint. The fact that your gut produces most of your serotonin has been known since the 1950s, when serotonin was first isolated from intestinal tissue. For decades, scientists assumed this gut serotonin was simply a local regulator of intestinal movement, unrelated to mood. That assumption was wrong. In 2015, a team at Caltech led by Elaine Xiao demonstrated that specific gut bacteria are required for normal serotonin production in the colon. Germ-free mice, raised in sterile conditions with no gut bacteria, had serotonin levels in their colonic cells roughly 60% lower than normal. When the researchers reintroduced spore-forming bacteria from normal mouse guts, serotonin levels returned to normal within weeks. The mechanism involves short-chain fatty acids, metabolic byproducts of bacterial fermentation, that stimulate the gut's enterochromaffin cells to produce serotonin. Your serotonin supply is not simply a function of your genes or your diet. It is a collaborative output of you and your microbiome. The primary communication highway between your gut and your brain is the vagus nerve. It runs from the brainstem down through the neck, chest, and abdomen, branching into the gut wall. What is remarkable is the direction of its traffic. Approximately 80 to 90% of the signals traveling along the vagus nerve run upward, from gut to brain, not brain to gut. Your brain is receiving a near constant stream of chemical and electrical signals from your gut. Much of this signaling is driven by bacteria. They produce metabolites that activate nerve endings in the gut wall, sending signals up the vagus nerve to the brainstem and beyond. In 2011, a landmark study by John Cryan and Ted Dinan at University College Cork, published in the Proceedings of the National Academy of Sciences, revealed something extraordinary. They fed mice a broth containing Lactobacillus rhamnosus JB1, a bacterium found in fermented foods. The mice showed dramatically reduced anxiety-like behavior in maze tests. Their stress hormone levels were significantly lower. When the researchers examined the brain chemistry, they found altered expression of GABA receptors, specifically the same receptors targeted by anti-anxiety medications. GABA is the brain's primary inhibitory neurotransmitter, the signal that quiets neural activity and reduces anxiety. The bacterium Lactobacillus rhamnosus was producing GABA directly in the gut and altering the brain's receptor sensitivity. Critically, when the researchers severed the vagus nerve, all anxiolytic effects disappeared. The pathway was clear: gut bacteria, vagus nerve, brain. A single bacterial strain, delivered through food, had chemically reconfigured the brain. Dopamine, the neurotransmitter most associated with reward, motivation, and craving, is also produced in the gut. Roughly half of your body's dopamine is synthesized in your intestines. Bacterial species including Bacillus subtilis and certain strains of lactobacillus produce L-dopa, the direct precursor to dopamine. Studies have shown that microbial composition strongly predicts dopamine metabolite levels in both gut and brain tissue. The practical implication is significant. Your cravings, your desire for sugar, fat, salt, may be partly orchestrated by microbial communities that benefit from those specific nutrients. Bacteria that thrive on sugar produce signals that increase the host's appetite for sugar. The host eats more sugar. The bacteria proliferate. This is not metaphor. It is a proposed mechanism supported by animal studies. The most powerful evidence for microbial influence on behavior comes from germ-free animal studies. Germ-free mice are raised from birth in sterile conditions, with no exposure to any microorganisms. They develop profound behavioral abnormalities. They are more anxious, more prone to stress-induced illness, and show altered social behavior. Their brains show abnormal development in regions that govern emotion and stress response. In 2004, a team at the Karolinska Institute in Stockholm showed that germ-free mice have an exaggerated stress response, releasing far more corticosterone than normal mice under identical conditions. But the most striking finding was what happened when bacteria were reintroduced. Colonize germ-free mice with bacteria from normal mice early in life, and the exaggerated stress response normalizes. Colonize them in adulthood, and the effect is reduced, suggesting a critical developmental window in early life when the microbiome calibrates the stress system. This window corresponds to early childhood in humans, the period when antibiotic use is most common and most damaging to microbiome diversity. In 2016, a team at University College Cork, led by Ted Dinnan, published a study that drew immediate international attention. They transplanted fecal material, and therefore an entire gut microbiome, from clinically depressed human patients into germ-free rats. The rats subsequently showed the behavioral hallmarks of depression: reduced motivation, increased passive coping, altered gut chemistry. Rats that received microbiomes from healthy human donors showed none of these effects. The depressive behavior followed the bacteria. This was the first direct evidence that human depressive symptoms could be transferred to another species through the microbiome alone. A 2019 study in Nature Microbiology by Jeroen Raas at KU Leuven analyzed the gut microbiomes of over 1,000 people enrolled in a large Belgian population study. 2 bacterial genera, Coprococcus and Dialister, were consistently and significantly depleted in participants diagnosed with depression. The depletion held across multiple countries, multiple ethnicities, and remained after controlling for antidepressant use. Coprococcus is a butyrate-producing bacterium, butyrate being a short-chain fatty acid that crosses the blood-brain barrier and has no neuroprotective and anti-inflammatory effects. Its absence may not be a consequence of depression, it may be a contributor. The gut-brain connection extends to neurodegenerative disease. Parkinson's disease is characterized by the accumulation of a misfolded protein called alpha-synuclein in neurons. For decades, Parkinson's was considered exclusively a brain disease. In 2003, German neurologist Heiko Braak proposed that Parkinson's might actually begin in the gut. He observed that alpha-synuclein pathology appears in the enteric nervous system years before it appears in the brain. The misfolded protein, he hypothesized, travels from gut neurons up the vagus nerve to the brainstem, then spreads through the brain. A 2019 study published in the journal Gut found that patients who had undergone vagotomy, surgical cutting of the vagus nerve, had a significantly reduced risk of developing Parkinson's over a 20-year follow-up period. The nerve that carries microbiome signals to your brain may also carry the earliest seeds of neurodegeneration. In 2013, John Krein and Ted Dinnan coined the term Psychobiotics, bacteria that, when consumed in adequate amounts, produce a measurable benefit in patients with psychiatric conditions. This was a radical proposal, that mental health could be treated, in part, by targeting the gut. A 2011 randomized controlled trial published in the British Journal of Nutrition found that Lactobacillus helveticus and Bifidobacterium longum supplementation significantly reduced self-reported depression and anxiety scores, and lowered urinary cortisol levels in healthy volunteers. Diet is the most powerful lever available for reshaping the microbiome. A 2021 study in Cell, led by Wastick, Sonnenberg, and Gardner at Stanford, compared a high-fiber diet against a fermented food diet in healthy adults over 10 weeks. The fermented food group showed significantly increased microbiome diversity and measurably reduced levels of 19 inflammatory proteins associated with chronic disease and mood disorders. The changes occurred within weeks. Your breakfast is influencing your brain chemistry by determining which bacteria dominate your gut. The most dramatic clinical demonstration of microbiome manipulation is fecal microbiota transplantation. FMT involves transferring processed stool from a healthy donor into a recipient's gut, replacing the existing microbial community with a new one. For recurrent Clostridioides difficile infection, a deadly gut disease that antibiotics frequently fail to clear, FMT achieves cure rates above 90%. Researchers at institutions including Amsterdam UMC and the University of Auckland are now investigating FMT as a treatment for depression, autism spectrum disorder, and Parkinson's disease. The gut microbiome is no longer a background feature of your biology. It is an active, chemically sophisticated system that directly shapes your emotional and cognitive life. Here is what all of this means for you personally. The anxiety you felt this morning, was it yours? The craving you had an hour ago, was it a choice? The low mood that has followed you for years, is it a neurological fact, or is it a microbial signature? These are not rhetorical questions. They are the questions that researchers at major institutions are actively pursuing in clinical trials right now. The concept of personal responsibility for mood and behavior looks different when you understand that the chemistry producing that mood is partly manufactured by organisms you did not choose and cannot see. Your microbiome was shaped by how you were born, whether vaginally or by cesarean section, since birth mode is the single largest determinant of early microbiome composition. It was shaped by every antibiotic course you have ever taken, every dietary pattern, every person you have lived closely with, because intimate cohabitation causes microbiome convergence. The bacteria in your gut today are not the bacteria that were there 5 years ago. You have been carrying different microbial chemists, different serotonin producers, different GABA synthesizers, at every stage of your life. You were not the same person neurochemically at each of those stages, not only because you changed, but because your microbiome did. There is something deeply humbling in this. You are not a single organism navigating a world alone. You are a superorganism, a community of trillions, moving through life as a collective. Some of your thoughts have bacterial authors. Some of your fears have microbial roots. Some of the calm you feel after eating fermented food is chemistry, real, measurable bacterial chemistry, altering the activity of your cortex. The boundary between you and the ecosystem you carry is not where you assumed it was. It may not be a boundary at all. Your gut is not a separate system you occasionally remember to feed. It is a co-author of your inner life. You are the ecosystem.